Morning, everybody. Thank you so much for that lovely introduction, Laszlo, and for inviting us here to participate in this new initiative, trying to bring everyone together to tackle a problem as difficult as brain cancer is incredibly important because none of us can tackle it alone. And this is something that's really going to require a really great team to attack. So I'm going to speak to you today about one of the three programs that are running in SingLab. And what we do in SingLab is we've been taking a basic and translational approach to brain cancer since 2007 when I opened my lab there. And really at the same time as I was opening my lab, I was setting up a pediatric neurosurgery practice, running back and forth across the bridge on the fourth floor that connects McMaster Children's Hospital to the beautiful new Michael DeGroot Center for Learning and Discovery, which is just behind, facing the university campus. So I kind of see that physical running across the bridge, sometimes carrying a brain tumor specimen over to the lab, which, by the way, my former supervisor, Peter Dirks, used to do with me when I was in the lab, also connected by a bridge at the Brain Tumor Research Center, where I'm lecturing next week, and everything comes full circle, you see. But there was also a bridge there. But I think that bridge really epitomizes the sort of life of someone who wants to be a clinician scientist, who wants to apply their life's work to one problem. And that bridge connects the hospital to the lab. And in the hospital, I'm often faced with dealing with patients with intractable or incurable types of brain cancer. And we try everything possible, even heroic things on the medical side to try to fix it. And we come to the end of the rope at some point, and we realize we have nothing else to offer. And that's the point where I cross the bridge and go to the lab. And then I tell everyone in the lab, this is an insolvable clinical problem. Can you tackle this? And of course, there are all these young, energetic, incredibly brilliant students who are all like, yeah, let's get on that. And then that gives me energy to go back and deal with the patients on the other side. So that iteration, that bridge, that physical bridge is actually the bridge that I think represents translation. And I think that would be the goal of the team, is to translate our basic discoveries into solutions for patients who currently don't have solutions. And one of those patient populations who currently doesn't have a solution is the patient with glioblastoma. Because this tumor, and I'll just quickly share my disclosures. I'll come back to this actually. But the patients with glioblastoma actually face a 5-year survivorship that is only 4% in Canada, 5% in the US. So that means 96% of patients, when they're diagnosed with glioblastoma, are fully aware that the end of standard of care, which consists of gross total safe maximal surgical resection, followed by cranial irradiation and temozolomide, they are aware that 96% of them are going to relapse. And the unfortunate thing is that when they relapse, there's actually no good second-line standard of care. And so the standard of care for a relapsed GBM patient is enroll in a clinical trial. And so you can imagine when you're diagnosed with glioblastoma and you look at your future, you're looking at a median, we've eked out a 17-month 5-year survivorship, a 17-month median survivorship, and really not a lot of ways to guide someone through that minefield of, well, which clinical trial should I enroll in? So these are all the problems that are faced with GBM, and the biology that underpins that clinical dilemma is here on this slide. And so what I'll share with you is just that this is the heat map of the original TCGA classification, which was done in the sort of late 1990s, early 2000s, pioneered by Roel Verhaak. And this is our very first stab at molecular classification, because of course, the GBM was the first tumor that the TCGA decided to perform whole exome sequencing and sequencing on. And so looking at over 400, 500 originally samples of GBM, we came up with this molecular classification. And please ignore the neural, because that has fallen out. People realize that GBM diffusely infiltrates and invades the brain, and we're just capturing a brain signature there. But if you look at the three molecular subtypes, proneural, classical, and mesenchymal, you would think, okay, that's great. If I have a classical GBM, which is usually characterized by EGFR variant 3 mutations and EGFR amplifications, why not put me in for a targeted EGFR therapy, right? There's so many out there. There's EGFR ADCs, there's EGFR CAR-Ts, that should work. Theoretically, that should work, but it doesn't in practice. And in fact, unlike breast cancer, none of our molecular subtypes in GBM are either predictive or prognostic. And the reason for that is the unbearable degree of heterogeneity that exists at the cellular level, at the genetic level, at the epigenetic level, and at the functional level. And I'll tell you about that briefly. So the way we begin to understand the heterogeneity is in the mid-2000 teens, when people started to do single-cell sequencing and deconvolute the complexity of GBM. So in this first hexagon, imagine that you're a patient who at primary tumor resection has been labeled with a classical GBM. And you'll notice here that only about 40% of the cells in this hexagon are actually blue. And that means that it's really just a dominant molecular subtype, but there are actually cells representing all of the molecular subtypes in this GBM that is called classical. And in fact, even more frighteningly, see the black dots? Those are unclassified. So even our molecular classification is not capturing all of the cells within a given GBM. And now you put that patient through the therapeutic selective bottleneck of standard of care, and you drive those cells to form different, distinct clonal hierarchies that then evolve through time and through therapy to give rise to a tumor here at relapse that is entirely a completely different cellular and genetic composition from the original primary GBM. So your GBM has aggressively and rapidly evolved through time and therapy to turn into a different tumor. And at that time is when patients are enrolling in clinical trials. And they're enrolling in clinical trials based on the biological information that we got from the first tumor, because only 20% of patients worldwide undergo a second surgical resection for GBM. So that means we really don't have a biological view of the relapse, and we don't understand it in the majority of patients. And so we go back to the primary biopsy and say, that's okay, we'll treat them based on this. But you can imagine that 63% of the clones of the actionable driving targets in the primary GBM have actually evolved out of the recurrent GBM. So you're almost dealing with a different tumor. And furthermore, functionally, the heterogeneity is kind of appalling. What you see here is a heat map from the favorite paper of my former supervisor, Pediatric. And you can see this heat map is from a paper he published in PNAS, where he did an elegant experiment where he took single GBM stem cells from a single GBM patient and plated them as colonies on the x-axis. So each of these grew into a tumor sphere. And then here is a panel of known chemotherapeutics that we tend to throw at every solid cancer. And what you can see is that every single clone from the same patient has a differential drug response. So that amount of heterogeneity is incredibly overwhelming. So this is what I usually cross the bridge and give to my students and say, here, deal with this. But what I will tell you is I'm going to go back now and just give you a tiny little bit more background about what we do. So why did we set up the Center for Discovery and Cancer Research at McMaster University? In 2020, the institute to which I was recruited, the Stem Cell and Cancer Research Institute, was dissolved. And in its footprint, we had this beautiful 22,000 square feet of open concept communal lab space, beautifully designed, amazing lab space. And the university said to me, what could we create in this footprint? And it was a wonderful question. Unfortunately, I know McMaster very well. I went to med school there, and it's an amazing place. But the key thing I wanted to do in building the CDCR is try to figure out what were our foundational and distinguishing strengths. What do we do at McMaster University that you could arguably say we do better than everyone in Canada or maybe even globally? And so what we came up with here is we were kind of early adopters in the field of immuno-oncology. Many of you know the virologist Frank Graham started his career at McMaster University, trained out many amazing people, many of whom did an offshoot from virology into viral oncology, and started developing oncolytic viruses. Then Jonathan Branson came along in the T-cell space, and I started investigating immunotherapies for brain cancer in 2015. So we built quite some momentum in that area. And furthermore, we seem to be good at innovating and bringing those discoveries to commercialization. So immuno-oncology is one area of focus, and 6 out of our 10 investigators have expertise in immuno-oncology. Secondly, radiochemistry. For those of you who haven't visited McMaster, please come and visit the CCR, and you will see that looking out of one of our south windows is a nuclear reactor. And that's because we make all the radioisotopes for all of the cardiac studies in all of Ontario. And we have amazing radiochemists like John Valiant, who set up the CPDC, the Center for Probe Development and Commercialization. John is an amazing person who was, again, a very early adopter to think about theranostics and molecular probes. And if I label HER2 on a breast cancer cell and I switch out the chemical linker, I can now attach a payload and turn it into a therapeutic. So he's been doing that for some time and building great strength at McMaster University. And finally, what we're known for worldwide is clinical epidemiology and evidence-based trials. And so these things all fit very well together for a cancer biologist who wants to discover something that they want to see translated into patients. And so we're very lucky to welcome Hong Han, and even luckier as well that Hong will soon have good company in Katie Houlihan, who will be our 11th PI. And she is fantastic. She's did her PhD with Paul Boutros. Now she's doing her postdoc with Christina Curtis, and she's studying germline variants and mutations in cancer and trying to understand how germline mutations may drive cancer risk. So it's a very exciting place. Please come and visit. And the Singh Lab is housed within the CDCR. And as I mentioned in Singh Lab, we have 3 teams, and we try to tackle the worst version of 3 prevalent brain cancers. The first team is adult glioblastoma. And as I mentioned, we don't have very good models of treatment-refractory recurrent glioblastoma. And so that's what that team focuses on. We have a team medulloblastoma who focuses on mostly MYC-driven group 3 medulloblastoma as well as group 4 medulloblastoma, simply because those are the 2 subtypes that are most prevalent at recurrence, and we want to deal with the unsolvable problems. So with medulloblastoma, we have a 5-year survival rate of 80%, which is fantastic. But for those 20 or 30% of children who still relapse, we have almost nothing to offer them. So that's what that team focuses on. And finally, team brain metastasis. Brain metastases, or secondary brain cancers, are 10 times more common than primary brain cancers, and becoming more common as we generate more cancer survivors through all the incredible work done by scientists at OACR for lung cancer and prostate cancer and breast cancer. We're actually generating people who can go on to get brain metastases. And this is a really difficult problem to solve, and my team is taking more of an intersectional approach. How can we block the brain metastasis initiating cell from getting to the brain in the first place? So just a little bit about all of that. Coming back to GBM, as I mentioned, there's really not much that you can do alone these days. Science is a team sport, and the most rewarding and incredible work that I get to do as a scientist is always within a team. So this is a team that we assembled around a Terry Fox program project grant, New Frontiers program project grant. And the idea is that we wanted, recognizing that the majority of therapeutics that we have designed for GBM fail, and there are multiple and pleiotropic reasons for that failure. But one of the reasons is, as I mentioned in the sort of evolution story, we're not hitting the right targets at recurrence. We don't know the right targets, and our drugs don't hit them. So part of the incentive for this program was to design beautiful models of recurrent GBM in Singh Lab, and then have this team of incredibly talented scientists help to probe the landscape, genomic, epigenomic of recurrent GBM, and try to find new actionable targets. And then we would design immunotherapies against those targets, and then go back into the same models and test them again. So the work that I'm going to share with you today is the work of very bright students in Singh Lab, as well as bright students and PIs from this entire program. So we already talked about how GBM evolves through therapy. And what we discovered when we dropped a barcode library from the Moffitt lab on the very beginning cell, the beginning of brain cancer, and then watched it evolve in our immunocompromised mouse models through therapy and to the end, is we discovered that sadly, every GBM has a different path to recurrence. And furthermore, we had no good mathematical way to predict the path to recurrence from the biopsy of the primary GBM. And that makes life extremely difficult, especially if you think about the fact that here on the top, you have a more predictable cancer stem cell model of GBM, where there is a normal cell of origin that is transformed into a tumor-initiating cell, and then that gives rise to clonal progeny through asymmetric division, and that cell is able to evade therapy and go through therapy because of very distinct mechanisms that they're radioresistant and chemoresistant, and then drive the final tumor. And the good news about this type of hypothesis is that you can actually find the cancer stem cell population in the original tumor. It's rare, it's like a needle in a haystack, but theoretically it's there. So you could actually treat that type of GBM. But these types of GBMs are not treatable. And the reason is that if you evolve from a tumor-initiating cell, but then a different clonal population takes over and then acquires a mutation through therapy. After all, temozolomide is an alkylating agent. it derives a hypermutation phenotype. In fact, the GBM here at this point has a much higher mutational burden than over here. And so if you're acquiring a mutation at this point, and then that is the driver that goes on to drive the recurrent GBM, you can never find this mutation or cell population in the primary biopsy. And so the problem is, for these different modes of recurrence, there's no way to really understand unless you can better understand the biology of the recurrent GBM. And I like to quote from this beautiful book by Dr. Henry Marsh. If you haven't read it yet, it's called Do No Harm, and it's a really beautiful compilation, not of all of his successes as a lifetime top neurosurgeon at University College London, but rather he reflects on his failures. Every chapter tells about a patient that he couldn't save or that he couldn't fix, or that in some cases he hurt. And that's what we have to deal with as surgeons. So there's this beautiful quote at the beginning: every surgeon carries within himself a small cemetery where from time to time he goes to pray. So we carry our failures with us, but the importance of that is not just to make us feel horrible all the time, it's so that we learn from them and that we bring that learning to the next patient. And so you could argue that glioblastoma, as my friend Warren Mason might say, is a graveyard of clinical trials because there are so many failed clinical trials. We only have 5 approved drugs. And the last breakthrough was temozolomide, which is part of the standard of care, and that was in the early 2000s. So we do not have a good track record, and the big drop-off is at phase 3. We can't get anything into or past success succeeding in phase 3. And so the problem is that you could say that these are arguably due to solvable problems, these failures. So if you look at the bottom point here, this is the point that I'm trying to share with you, is that we really aren't looking at the right targets. The targets that we design drugs against are relevant in treatment-naive patients, but they're selected against following frontline therapy. Also, this is a big problem that we have to deal with. There are very few companies. As you guys all know, this year has been terrible for biotech, and everyone's saving their lead program, but all of the other programs are getting cut. And I remember talking to Carl June about his company, Unity, where he was designing amazing CAR T cells. And he told me that GBM was the 11th program. And he was like, oh boy, that's not good news. That's going to get cut. So we need more focus by pharma as well on this terrible brain tumor. So why immunotherapy? Why would we choose this for glioblastoma? So if you think this is a review that we wrote and published about these 3 classes of new immunotherapeutics. And what we did is we surveyed all of the trials that have happened in these areas against every known target in these 3 different types of immunotherapies over the past decade, and review them and try to apply that approach of understanding the failures of these trials. Because I've mentioned to you many times now that these trials all fail. But the idea is not just to throw the failure behind you and walk away. What can we learn from those failures? And very often there are silver linings where there's always a tiny subgroup of patients, as I've learned from my clinical epidemiology colleagues at MAC, that are responders. And they're buried in the text of the overall failure of the trial. So go and look at those people and try to figure out, well, why did they respond to this therapy? That's another thing that we don't tend to do as scientists. And here is an example of something that is greatly advancing new immunotherapy approaches for GBM. This is from my friend Christine Brown at City of Hope in LA, who's an amazing CAR T cell scientist who works with a neurosurgeon called Ben Abadi. And together they published this paper in the New England Journal of Medicine that was, I think, a major game changer. So what they did here is they showed that they could treat patients with their IL receptor 13 alpha 2 CAR T cell, and they could deliver it straight into the brain, and they could put it into the cavity of where the brain tumor was resected, and that could cause regression of the tumor, and that's awesome. But unfortunately, what happened there was antigen escape, and that antigen evolved out of the tumor, and then the CAR T was no longer effective, and that was bad news. But this one poor patient, this was an incredible N of 1 first-in-man study. They delivered this first IV, then they delivered it into the tumor cavity, and then they delivered it into the ventricle. So when you deliver something into the CSF, something that is already a homing missile by nature, because that's what CAR Ts are. They're homing missiles and they find their target no matter where they're delivered. But if you deliver that into the CSF, you have the additional benefit of circulating fluid that will take your therapy around the entire neuraxis and deliver it everywhere the GBM might be. And so when they did this, they actually found that with intraventricular delivery of IL receptor alpha to CAR T cells, they actually found regression not only of the local tumor, but also of the tumor in the opposite hemisphere, where the GBM, of course, likes to travel across the corpus callosum and then invade the opposite hemisphere, which is rarely surgically treatable. And then it likes to disseminate around the left meninges of the spine. So the GBM spreads rampantly. And when you deliver the therapy into the CSF, it gets rid of the tumor, original site, other hemisphere, and even the spinal nodules receded. Unfortunately, the patient did succumb to antigen escape in the end because the target wasn't good. But this mode of delivery was remarkable. So local regional delivery for brain cancer, if you can't get a drug across the blood-brain barrier, just bypass the blood-brain barrier, go straight into the brain. So what are the lessons that we've learned? Some of them from all of these GBM treatment failures. So these are the challenges that I think we have to deal with. So we've realized that we have to better understand the biology of GBM recurrence. Why? Because that's the time point at which patients are enrolling in clinical trials. If you want your therapeutic to work, study the tumor of the patient that you're actually treating. Monotherapies are not going to succeed in such a heterogeneous tumor. We are going to have to design therapeutics that are going to attack all of the pleiotropic signaling pathways active in all of the heterogeneous GBM cells. So rational combinatorial polytherapies. It would be best if this could be a data-driven approach. Could we somehow analyze that minimal residual disease stage and figure out what's there and then design therapies based on the patient's own data? The therapeutics have to target, as I'll get to shortly, not only the GBM cells, but also the cognate tumor immune microenvironment. There's a very immunosuppressive niche created by the immune infiltrates around GBM, and we'll talk about that more. And finally, local regional delivery can actually promote better trafficking and durability and persistence of cell therapies, and maybe an ideal way to get to brain cancer. So how did we deal with the problem of we don't know much about the biology of recurrent GBM? In SingLab, we decided to build these dynamic models of cancer. And this is what I've been doing since 2007 when we first set up the lab. Cancer is not a static disease. It doesn't help to have one biopsy or one snapshot. You want to model it through time, and if you can, through therapy. So here we adapted the SHIP protocol, which is the standard of care for GBM, for the metabolism of immunocompromised mice. Not that easy. My vet was not my best friend when I started at McMaster because she said, you want to treat immune-compromised mice with chemo and radiation after you operate on them? Are you crazy? So this took a long time to optimize, but once we did optimize it, what we got was a model that looks very, very much like humans who recur with GBM. So the mice get a benefit from their radiation and temozolomide, and they do get a survivor benefit, but about the 2 weeks in a lifespan of a non-skin mouse matches about the 5 to 8 months of a honeymoon period between when a patient finishes standard of care and when they relapse. It's about average 8 months time. So this model was good, and when we surveyed those relapses and compared them to the 20% of recurrent GBMs that we operate on, there was a good match. So this is a faithful and reproducible model that recapitulates what we see in human patients. And this is where all my colleagues came in. And in fact, it was Jason Moffat who visited my lab in 2015 and said, hey, I heard that you have some really good models of recurrent GBM. And I'm wondering if we could study them. And I'm also wondering if you could test out this new antibody I made in your models. And he came in carrying a vial of RW03, which is the antibody he designed against CD133. I'll get to that shortly. But regardless, the purpose of this pipeline project was in project 1, we would try to carry out an integrated multi-omic analysis of this beautiful PDX model that we developed and analyze at every stage of the model the different genetic and genomic composition of the tumor. And then we would try to distill what are the targets that are most enriched at recurrence. Then we would go on to our amazing medicinal chemists and antibody engineers. We were actually now agnostic to the therapeutic. We like immunotherapy, but hey, you know, Jake McGolan, if you have a small molecule for me, I would love that too. So whatever our targets are, we would design an appropriate therapy against that and go back into the same models that served as a discovery platform. Now they're a preclinical testing model. And this way we were able to generate very, very nice preclinical data packages and try to entice those people in industry to make more drugs against GBM. And here's just a schema of what we did in project 1, which was led by Jason. And you can see here on the left, we tried to mine all of the models we have. So here we have our PDX adapted model. And here we have the actual matched primary and recurrent pairs of tumors that we get from the actually 27% of patients who undergo surgery for recurrence at Hamilton Health Sciences, where I have very aggressive neurosurgeons who like to operate at recurrence. That's very lucky for, it turns out, for the patients and for us. So we capture the biology of the primary and recurrent matched pairs and interrogate those as well. And what we've done here is barcoding. You can see every stage of the model. So this is when we first extract the tumor, and then we characterize the very short-term culture of the brain tumor initiating cells. They're engrafted into the frontal region of the non-skin mouse or the NSG mouse. And then we treat them with the strip protocol. There's this fascinating phase where the tumor regresses. There's this debulking of the tumor. And you can see the MRI of the mouse at that time. Sometimes it looks negative, but we know there's 100 to 3,000 cells that are hiding in the brain at that point. They're tagged with GFP, so we can remove the entire mouse brain. And it may look histologically negative, radiologically negative, but when we dissociate that brain and we recover the GFP positive cells, those are the seeds, the reservoir of recurrence. And we call that MRD. We're imitating hematopoietic people because they've always blazed the trail before us. That MRD state is highly informative. And in fact, if you can capture those 100 to 3,000 cells from the mouse brain and profile them, you will find a signature that shows you the very cells that are going to generate that whole explosive clonal hierarchy again at recurrence. And here's recurrence. Then, of course, on the matched pairs, we've been undertaking glycocapture as well as whole cell surface proteomics with Dr. Thomas Kisslinger, and also genome-wide CRISPR screens of the primary and matched recurrent pairs from the patients. So this is the most difficult thing about this whole schema. And my students would be angry if you say that, because this was a huge amount of work. But the truth is this is the magic. And this is the difficult part. And this is where we turn to someone brilliant like Dr. Han, who can help us to try to figure out not just what do we do with all of this data. There's different platforms, different levels of data. How do we integrate it? How do we prioritize our targets? And so I think that's a large driving force of why biocomputational power is so needed to tackle these problems. And Hong and Katie will probably be the most important people on our team. So just very quickly take you through the platforms that we built. Here is our DNA barcoding. This was done by Maliha Kazi, who completed her PhD in my lab. Now she's a first-year radiation oncology resident here at KNH. And Maliha published this beautiful paper in Cell Reports that tells you essentially what I mentioned before, is that all GBM patients recur differently, which is really disappointing. And this, of course, is our CRISPR-Cas screening. And I want to show you that this is really alarming. Here, this is the primary GBM and the recurrent GBM from the same patient. Look at the vast difference in the essential genes that come out. This was a CRISPR knockout screen we did, and you can see there are very, very different drivers of recurrence. And this work was done by Charlie Ashokshi and David Chu in the Moffitt lab, and it's under revision right now. I hope you'll see it soon. And we published this in Acta Neuropathologica as a tour de force by Nazanin, my PhD, who's now doing a postdoc in Switzerland, and Shabazz Khan from the Kisslinger lab. And here Thomas did proteomics on 136 GBMs, and 40 of those were matched primary and recurrent pairs from the same patients. And we learned a lot about the signatures, and please do the paper. This is just an example of a patient. So here you see one patient, the primary GBM represented in the red and the recurrent GBM in the blue. And I want you to notice that this patient was classified as a classical GBM because look how high EGFR is on the volcano plot. And then you can see that there's no EGFR at recurrence. So this is a patient that we actually modeled in our mouse model. And the patient was enrolled in the ABT414 study, which was an ADC against EGFR. And so it's possible that she could have gotten an EGFR drug at this point. And perhaps that's why that clone is now gone, because it was successfully eradicated by therapy. At the end of the trial, the patient called AbbVie and said, can you tell me where, was I randomized or was I placebo? Was I randomized to treatment or was I placebo? And it turned out she was placebo. So you can see that that clone vanished on its own just through clonal evolution. So the other great thing is we did call AbbVie, who sent us the same drug that the patient was taking in the trial, so we could run a trial in her mouse xenograft. So that was really exciting. The patient would sometimes call me and say, how are my mice doing? So the other thing that we picked up in the landscape of recurrence is this massive difference in immunosuppressive marks. And remember, we're interrogating the cell intrinsic pathways of the GBM cell itself. We don't have immune cell infiltrates represented here. This is just the B-ticks. So the B-ticks themselves are sending immunosuppressive signals to the niche. These GBM cells are sending signals to tell macrophages to quiet me down and become M2 and don't eat me, you know. So it's really frightening to see the signals that the GBM cells are expressing, and here are the targets. I'm going to leave this all to Hong, but our last platform she's going to tell you all about. We have this very elegant, beautiful single cell sequencing platform. The thing about Hong is that she builds the technology and then she builds the analytic pipeline. So the most exciting thing about this is really the analytic pipeline. So this is really beautiful, and Nick Mikolajewicz, a very talented med student in the Moffitt lab, actually has made a computer version of this where you can go and look at publicly available as well as our matched primary and recurrent GBM pairs, and you can just patrol the site and look for your targets of interest and see where they crop up in the single cell data, and it's really beautiful. And what we've really found is we've learned a lot, not only from mouse models that we've set up, but from these matched primary and recurrent GBMs where we can learn about the immune cell infiltrates as well. And in fact, this is one of my favorite things. We're just putting through a paper with Nick where he has found a really exciting ligand receptor interaction between a GBM cell and a brain cell in the niche. And so, and it's an oligodendrocyte. And there's a very interesting signaling pattern happening between those cells. And this is kind of the whole field of cancer neuroscience, which is very hot right now. So I told you about the platforms we developed in our program. Now I'm going to tell you about some of the targets, because I'm sure you're all wondering, okay, you told us that you found all these new targets in recurrent GBM. What are they and what did you do to target them? So the first translation case study I'll tell you about is one that you probably have all heard me talk about before. CD133 is the original marker that I used in the lab of Peter Dirks to isolate brain tumor initiating cells. And you can see that this is a very strong marker of cancer stem cells across multiple solid cancers. And in fact, it's practically a marker of treatment resistance because it is almost always highly enriched in patients with treatment-refractory recurrent cancer. And so what we did is we decided to rationally develop a whole spate of immunotherapeutics against our W03, the beautiful antibody that Jason made. And what we did here is we tried a naked antibody, we tried a bispecific T-cell engager approach, and we tried a CAR T-cell approach against the same target, our W03, in the same models. And so what we found is the CAR T-cell won. That's the short tagline. And again, I think it's because of the Christine Bonan story. I think it's because that CAR T is like a homing missile. A bite is like a small molecule. It's going to diffuse across the chemical gradient. At some point, it's going to engage its T cells in the niche, but there aren't that many T cells in the GBM niche, which is part of the problem with the bite approach. But furthermore, at some point, it's not able to penetrate or get around the solid tumor or find all of the infiltrated cells that cross the corpus callosum and invade the opposite hemisphere. But a CAR T cell can. They're like homing missiles. So we were able to spin out a small company from the Terry Fox program called Empiric Therapeutics, which was acquired by a big NASDAQ index company, Century Therapeutics, who promptly made GBM one of its programs. So that was a win. We have encountered hurdles since then in this biotech climate, but we push on. So you can see here the results of the CD133 CAR T. In a tumor that expresses 90% CD133, you get 90% killing. And so this is a highly specific and sensitive antibody, and the CAR T cell works incredibly and exquisitely well. And fortunately, we were able to include in this paper a very elegant model, and this with help from my colleague Kristen Ho. We generated basically a humanized mouse model where we injected human cells into the bone marrow of immunocompromised mice, and then we were able to treat those mice with our different therapies to find out, are we targeting other CD133 positive essential cells like hematopoietic stem cells? And so in this elegant toxicity model, we were able to show that in fact, our treatment does not significantly reduce numbers of human hematopoietic stem cells. And there was a CD133 CAR T trial done at a hospital in China that showed essentially the same thing as our models. The mice would develop anemia that would self-recover. The patients did the same thing. And in fact, the great thing about the program and having the 3 different programs is that there's iterative crosstalk between the teams. And the students aren't isolated in little silos. They all work together and they love talking to each other. So the people who did the CD133 CAR T paper talked to Agatha, who is a PhD student, now a postdoc on the brain mets team. And Agatha said, well, I'm going to go check my brain mets and see if they express CD133. And indeed they do. So we went and took the CD133 CAR T and trialed it in our brain mets models. And we found an incredible response, especially in one type of brain met. And I need to tell you this story because it's an homage to Catherine O'Brien. So what you see here is the results of the CD133 CAR T in our colon to brain metastasis model. Yeah, this is crazy, right? Where the mice are cured. So we looked at the brains and this obliterates them. So you guys may remember many, many years ago, Dr. O'Brien discovered a CD133 positive colon cancer initiating cell. And in fact, I think that's what this CAR T is targeting in colon cancer. And so this was an incredibly surprising result, and Agatha was so thrilled, and this paper was just published in Clinical Cancer Research, and our colleague Justin Mathia wrote this punny editorial about it, and so it's gotten a lot of attention. So yes, CD133 in the driver's seat, and we think that this could be actually a very good second indication for the CD133 CAR T cell. And in fact, Agatha has carried her love of drug discovery now over into small molecule work. And she's doing this very elegant work now that was actually funded by Admiring Therapeutics. And again, working with my interdisciplinary colleagues, Jason Magdalene is an amazing medicinal chemist at McMaster University. And we've been able to find a target in our brain mets models that permits us to try to intercept that cell and prevent it from getting to the brain. And so hopefully I can come and give another talk to you about this in the future. Translational case study number 2. I want to tell you the story of GPNMB. GPNMB was a project taken on by Neil Savage, who's a wonderful PhD student who's now at Dana-Farber. He's a scientist there. And what he did is he looked at the old data of my student Parvez, who when he was working on a CD133, did this lovely sorting experiment where he took normal human neural stem cells and GBM stem cells, or rather cells from the GBM, and sorted them into different populations. So here you can see you have normal neural stem cells that are CD133 positive, GBM stem cells that are CD133 positive, and then there are CD133 negative non-stem cell compartments. And what we found is that GPNMB was actually the most statistically significantly differentially expressed gene in the CD133 negative GBM cell population. Sorry. And so in fact, he found that the GPNMB transcript levels were very low in the normal neural stem cell populations, which is important. And what we saw when we went and looked at our proteomics is that GPNMB is highly enriched at recurrence. And in fact, these are the 40 patients that Thomas profiled, the matched pairs. And most excitingly, when we go and look at the brains of the mice who were treated with the CD133 CAR T, remember I told you there were 10% of cells left over that are CD133 negative, and fortunately they're very indolent, so they're not reforming a GBM very quickly, but they're still there and they need to be targeted. Those cells are positive for GPNMB. So this suggests that perhaps there's a good therapeutic combination we could think about. GPNMB is already an immunotherapy candidate for multiple cancers, and I want to point out that this glycoprotein is, of course, with the name of non-metastatic melanoma protein B, is found in tumors that are mostly mesenchymal in origin. So GPNMB is highly enriched in fibrolytic osteosarcomas, in fibrolytic breast cancer, and my colleague Peter Siegel at McGill has studied GPNMB in breast cancer, and this has already gone to trial with an antibody called CDX-011 against GPNMB. Interestingly, speaking of cancer neuroscience, this is really interesting to know that there's a broader role and context for GPNMB in neuropathology that may be related to its role in cancer. So GPNMB was actually found in this paper that was published in Science in 2022, and it was a GWAS study. And what they found is that on the 7th chromosome, they found a gene that was basically linked to that was GPNMB, that was linked to a very, very high risk of Parkinson's disease. And they found that actually GPNMB is interacting with alpha-synuclein and is increasing its expression and causing cellular uptake of the fibrillar version that causes the neurofibrillary tangles. And it actually helps to assist in the development of the pathology in Parkinson's disease. And so you can see here that, that this really implies that GPNMB has a role not only potentially in the GBM cells, but maybe is doing something in normal brain cells or in the niche as well. So keeping that in mind, we thought from all of our studies that GPNMB would be a good target for recurrent GBM. And here we probed our proteomics and our tissue microarray, very kindly made by Dr. Cynthia Hawkins for my study. And you can see here that it really is highly expressed at recurrence. So we went to our wonderful antibody engineer, Dr. Kevin Henry at NRC, who made us a really good sensitive and specific binder for CAR T-cell engineering. And we found that our colleagues, Dr. Doug Mahoney and Frank Semp at University of Calgary, had already made a CAR T-cell, but using CDX011 as a binder. That's the commercially available in the public domain binder. So we thought, well, we'll do head-to-head testing with that one and with our antibody from Kevin Henry. And you can see here that both types of the GPNMB CAR Ts are highly efficacious as a monotherapy. But if you combine the GPNMB CAR T rationally, right, with the CD133 CAR T, there's actually an even better treatment response. We also, coming back to that neuroscience story, we went back to our single cell data and found that GPNMB is not only expressed in the GBM cells, they're highly enriched in the macrophages in the GBM niche. And that's not surprising because cells of mesodermal origin tend to express GPNMB. And so we realized when we went to our GL261 knockout mouse, we actually made a knockout of GPNMB. Sorry, it's not a knockout mouse. We CRISPR knocked out GPNMB in the GL261 tumors that we implanted into black 6 mice. And so what you can see here is that the GPNMB knockout actually mimics the improved survivorship we see in the mice treated with the GPNMB CAR T. So GPNMB is doing something important, and what we learned here is that it's actually influencing immune activation, because this is now an immunocompetent mouse model. Remember, the drawback of our PDX model is we cannot look at the tumor immune microenvironment. So we have to turn to syngeneic models for that. And in fact, it has long been known, this is a paper from Pam Sharma, and Dr. Sharma showed that actually there is a CD73 positive macrophage infiltrate that she found in GBMs of patients who had been treated and failed checkpoint inhibitors. I'll come to that in a minute. So these patients who were enrolled in a clinical trial, she got samples of their GBM, and she noticed that there was this population of CD73 VISTA1 positive and PD1 negative cells. And she found them through single cell sequencing. So she went back in a reverse translational approach to her lab and generated a CD73 knockout model and found that actually these CD73 high macrophages, these are myeloid cells that persist after anti-PD1 therapy, and they actually help to immunosuppress the GBM. And so she thought that we should be targeting these CD73 macrophages that are helping to immunosuppress the niche along with the GBM. So this concept of co-targeting the macrophage and the GBM, or the thyme and the cognate tumor, has been around. And in fact, my colleague Amy Heimberger did this beautiful window of opportunity trial, which I know Laszlo does too and loves. And you can see here the reason that the checkpoint inhibitors are not working in GBM patients is because there's actually very few T cells at the recurrent GBM niche. The recurrent GBM itself can be made in cellular composition, can be made up of up to 50% macrophages. So the macrophage is the dominant cell at recurrence. That's why it's not really worth targeting the T cells at recurrence. And in fact, Steve Pollard, my colleague, found that this is also frightening. It's not only those macrophages, the way that the GBM cells are evading therapy is they're acquiring a myeloid-affiliated transcriptional program. So in other words, they're masquerading as macrophages, and that's how they're evading therapy. So clearly we need a therapy that targets a macrophage phenotype. So fortunately, Sean Graywell, my new MD PhD student, wanted to follow up on Neil's work. And he said, I got to figure out a way to target these GBM-associated macrophages. And we used to have this binary model where we call them M1, M2, but now we're moving towards more the spectral model. So it's not as simplistic as just these are pro-tumorigenic, these are anti-tumorigenic, these are pro-inflammatory, these are anti-inflammatory. It's actually more complicated. And so there's a lot of crosstalk between the actual GBM stem cells and these macrophages. And how can we position ourselves to really figure this out? Well, fortunately, we have two labs, biomedical engineering labs, labs at McMaster run by Ryan Wiley, who many of you may know joined McMaster, trained in the lab of Molly Scheuchstadt. So he set up this beautiful lab, does all kinds of medical engineering, as well as Boyan Zhang. So they helped us to pioneer with hydrogels this new GBM macrophage co-culture system. So working with Sean, you can see here this very beautiful model. This is a primary GBM from our lab, and this is a human macrophage cell line. But you'll be excited to know that Sean, who has a committee meeting next week, has fresh data that shows that he can actually do this with the same macrophages from the patient. So we've now found a way to get the macrophages out of the GBMs. And now we have matched human macrophages and patient-derived GBM. So we can do this co-culture with the patient's own tumor immune microenvironment. And you can see here, this is a video of when you knock out CD47, the don't eat me signal. Now these GBM cells labeled in red are being eaten by the macrophages labeled in green. So this will really help us to study those interactions that we need to study. And here Sean found a very high CD163 high cluster, just like Pam Sharma found the CD73 high cluster in GBM. Sean found a CD163 high cluster in our GBM, recurrent GBM cohort. And he wanted to look at some of the signals there. And we're actually finding quite a few genes that are influencing the behavior of these tumor-associated macrophages. So coming back to completing Neil's story, Neil said, okay, great, I have this combinatorial therapy. It works really well for GBM. I can target the GBM BTIG compartment with CD133. I can target the GBM non-BTIG compartment with GPNMB. And I also target the time. And then, of course, we can answer that question in the PDX model. So here we turned to a humanized mouse model, and this was a very nail-biting, terrifying, expensive experiment, but we engrafted the humanized mice who have this beautiful composition. These are non-EM cell mice, and they have a nice complement of macrophages. That was an important thing. And then we engrafted them with GBMs, and then we treated them with the GPNMB CAR-T. And we're just waiting now for the Lunaphore data to come back and tell us whether we have eradicated those GPNMB positive macrophages from the time as well. And that will be very, very exciting to know shortly. And who's doing the Lunaphore beautiful multi-parameter protein immunofluorescence for us? It's Amy Heimberger and Hinda Najm at Northwestern. Very talented scientists. And just to give you a little hint of some of that data, it's not analyzed yet, but I'm very excited. So you can see the kind of images that we get with this view. We can track all of our immune cell compartments, all of our GBM cell compartments. And what we noticed is when we treat with the GPNMB CAR-T, the CAR-T itself seems to be relieving some immunosuppression in the niche. So that's our first sort of view of the data, and it's very exciting. Here is another story. I'll tell these very quickly because I think I may be over time. I won't steal too much of your time. I'll be very quick now. This is another story we told where we generated a CAR-T against CD70. CD70 is a tumor necrosis factor alpha family member, and you guys know it's the only cognate ligand of CD27. And so we know there's an interaction of CD70 with T cells that express CD27, but it's long been recognized as an actionable target in recurrent GBM, shown by my colleague Joy Mitchell. And so here we show that the CD70 CAR-T works really well in recurrent GBM. But this is the exciting part. We realized, you know what, some of the CD70 CAR-Ts are killing each other, right? So to prevent fratricide, Jason Moffat helped us through a GAP grant, a Genome Canada grant, to knock out CD70 and the CD70 CAR-T, and that sort of decreased our rates of fratricide. And it was really nice to see CRISPR Therapeutics take the very same approach. When we published our CD70 CAR-T paper, they called me and said, well, we're doing a molecular basket trial based on your paper. Should we include GBM? And I was like, yes, please include GBM. So they've generated a beautiful CAR-T with, I think it has 12 edits, actually. And one of them is the CD70 knockout. So that was good to know that that approach was good. And here again, you can see that there are complex interactions in the time. And what we reported in this paper is that CD27 is not only marking T cells, it's marking a population of immunosuppressive macrophages as well. So there's complex interactions happening between the GBM cell and the cells in the time. And the idea is, can we design therapies that actually kill two birds with one stone? GPNMB, it kills GBM cells and it kills macrophages at the time. CD70 could be killing T cells and macrophages in the time as well as the GBM cells. So this double jeopardy phenomenon could be positioned really well if we can find targets that are expressed in both the tumor and the time. Okay, I think this is the last story. So this is Will Naitch, my PhD student, who was very interested to pick up the target we found enriched, which is PUR, or plasminogen-activated urokinase receptor, also known as UPAR. So you guys may know that urokinase is the cognate ligand for PUR or UPAR. And what happens when you activate UPAR is we actually think that there's a cascade of signaling, usually through ERK and SARC and RAS intracellularly, but there's also some activation of integrins. So the overall endpoint here is degradation of the extracellular matrix. And this is what happens when you're able to activate UPAR. And in fact, in breast cancer, we know that this activates EMT pathways, or rather blocks EMT pathways. And so we wanted to find out, we knew that UPAR was highly expressed in our recurrent GBMs. As you can see here in the volcano plot, it's highly enriched in recurrence. And in fact, it also correlates with poor prognosis, you can see here. And also, it seems to be selectively enriched in the GBM cells compared to normal brain. So again, it could present as a good target for us. So we found that CRISPR knockout of UPAR actually inhibits a lot of the GBM stem-like characteristics. And we saw UPAR expression is very high in our recurrent GBM PDX models. And in fact, UPAR knockout improves survival. So going through all of the steps to try to figure out, could this be a good therapy for GBM? So Will is working on this story. And of course, Kevin Henry made these very nice and novel UPAR-directed single-domain camelid antibodies that work very well in our CARs. And the CARs are actually really potently cytotoxic. So while all of this was happening, a very interesting other story came out from Scott Lowe's lab, where he actually generated UPAR CAR-Ts, but in a very different context. This was published in Nature Aging. And these CAR-Ts, in addition to killing GBM, are also senolytic, so they prevent aging. So we think this is pretty amazing. If this could be a CAR-T where you could prevent people from getting GBM, prevent them from getting old, then that would be probably a very marketable therapy. So again, Will, following from the playbook of Agata, and I told you about the crosstalk in my lab, was like, hey, wonder if UPAR is expressed in our brain mets. And so he did find, in fact, UPAR is highly expressed in the brain mets, but not in the matched lung primary. And this makes sense because one of the processes that allows a lung cancer cell to undergo the metastatic cascade and break away from the primary lung cancer is EMT. But the activation of the EMT signature happens later during the metastatic cascade. So it makes sense that it's not on in the lung cancers, but it's going to be on in the brain met. And so here, look at this. This is crazy high levels of UPAR in all of our brain mets lines from multiple models coming from melanoma to brain, breast to brain, as well as lung to brain, all of which are in the same lab. So Will came to me this week armed with this data, asking permission to trial the UPAR CAR-T in the brain mets models, and I said yes. So last story, just 2 minutes to finish this off, because I've told you guys a lot about temporal heterogeneity, but I have not spoken at all about spatial heterogeneity. And the fact is that you cannot expect a tumor as heterogeneous as GBM to have a spatial representation that is uniform. And in fact, that tiny biopsy that the neurosurgeon sends you doesn't actually represent the whole tumor. Is the therapy you're designing against that tiny biopsy going to work for the whole GBM? We don't know. And in fact, my friend Charlie Swanton published this beautiful paper in the New England Journal of Medicine. You guys may remember this paper. It kind of changed the way I thought about cancer. He first described intratumoral heterogeneity having a branched evolution pattern, and he showed subclonal events that were driving the different regions of this kidney tumor to break out and have a different pathological subtype. So here you see a kidney tumor in 12 different regions. Part of them, some of them are WHO grade 1, some of them are WHO grade 4. There's different pathologies across this tumor. There's spatial heterogeneity driven by subclonal genetic events. And we do know about GBM in the sense that there are investigators who have tried to examine spatiotemporal evolution of primary GBM. But the bottom line is the majority of what we know about GBM comes from a tiny biopsy. And so we're only focusing on the trees. What about the forest? So I told you guys that we have a study at McMaster Hospital where we acquire patient-derived brain tumor samples. And we've now acquired 1,700 samples since we started the lab in 2007. And this is Cindy, who had donated both her primary GBM to us, and then she went through standard of care, and her tumor recurred, and she had a second surgery, and we got her second specimen as well. We could compare them. And then we even took her blood, and we made CAR T cells from her own blood and tried them in her own brain cancer models. But she was a scientist herself, and she recognized, as she said to us, you're never going to fully understand my GBM unless you see it at death. And so I would like to donate my brain and spinal cord to your lab when I die. And so she pioneered the concept for our lab of donating your entire GBM, permitting us to study the entire GBM. And so this story, which is so wonderful, made the front page of the Globe and Mail, which my media department tells me is important. Apparently, the most important stories that get attention are on the front page or the third page, because when people open the paper, the third page is the next thing they see. But it occurred to me that no one looks at physical papers anymore, so I don't know if that actually works. Regardless, this got a lot of attention, and I started getting emails from GBM patients all over Canada saying, I want to donate my brain like Cindy did. They were all inspired by her, which was remarkable. So that gave birth to a program in the lab called the Spatial Heterogeneity Challenge. So with this GBM donor rapid autopsy program, we are now able to study GBM in its entirety, the whole tumor. And furthermore, if we are able to get the patients to our anatomy lab bequeathal program within 4 hours of death, we can retrieve live cells from all of the different regions that span the GBM and basically do multi-regional biopsies of the GBM at death, and derive cell lines that represent every different spatial region of the GBM. And then we can characterize those lines. So instead of only looking at that tiny little dot that the surgeon gives us, now we can look at the whole GBM. And these are the teams involved in the Spatial Heterogeneity Challenge. We have this wonderful anatomy lab bequeathal program led by Dr. Bruce Wayman. We have Singh Lab that comes to retrieve the samples and take them, whisk them right up to the lab to dissociate them and make multiple cultures. And we have our wonderful neurosurgeons who help to do the extraction and the harvesting of the brain and spinal cord. So this is the protocol for how we do the sectioning. We've established this to be very rigorous, and we do the same for every donor. And you can see here that this is how we mark and label all of the distinct areas of interest. So for every one of them, we slice them in coronally into thirds. 1/3 goes for cell line derivation, 1/3 is formalin fixed. 1/3 is fresh frozen. So this is going to permit all types of single cell analysis, keep Holly busy for years. And then, of course, the formalin fixed paraffin tissue can also be used for multi-parameter protein imaging. And so we're going to extract as much data as we can. We have now had 13 donors come through the program, and there's another 6 enrolled. And this is sort of what it looks like. We also collect CSF as well as all of the other parts of the brain, and you can see how we label all of the different regions of the GBM. And this is sort of what the processing of the samples looks like. It's a massive amount of work, and we need a pretty big team to do this. And here's the beginning of some of the analyses that we're starting to do. Very quickly, I just want to show you this super quick thing. This is the original location of the tumor, T1. It was right frontal. This is the part we captured from the distal contralateral subventricular zone, an area you would never survey in a GBM patient, ever, right? So what we did is we sent this to Marco Mera, who is part of the Spatial Heterogeneity Challenge. And Marco and a very talented postdoc, Veronique, generated the SN multiome platform from 10x, looking at also at single cell RNA and ATAC-seq done on their own in those 2 samples. And in fact, they're even able to now do a single cell mutational or CNV profiling. And this is the sort of punchline here comes right here. So this is the mutational profile of T6. That's the distal contralateral subventricular zone of the GBM patient. And so you can, what this tells us is that those cells migrated across the corpus callosum and invaded the opposite hemisphere. And this is their signature. So this is something that I, as a scientist or a neurosurgeon, never usually see. So this is very new knowledge, and we're very excited about the knowledge that this challenge is going to provide. So I will end there. This is just some of the sections we've been doing with that multi-parameter IF. This is the type of technology we're going to throw at the Spatial Heterogeneity Challenge. I'd love to tell you about it in a few years when we finally get some data. I will finish there, and thank you. Sorry for going too long, too much excitement, but it's such a pleasure to be here. Thank you.
So Hong is going to talk about sort of her work on RNA splicing. I think, and some of the computational tools that she's used to study that, and how this is involved in brain development related to cancer. So,
Thank you for the invitation and also the kind introduction. It's really a great honor to be here today for this amazing program and visit. So I'm excited to discuss some of our recent work and also importantly, what we can achieve more together as a team. So cancer is the leading cause of death in Canada, and the numbers shown here are devastating. However, thanks to the effort of a large team, including many people here, clinicians, scientists, and other key parties, and so we can make a change and bring hopes. So for example, the survival rate have doubled in the past a few decades. So with the emerging new technologies and also evolving cancer research and management teams, it will be also important to see and work together as a team what we can achieve in the next 5, 10 years or 50 years. So cancer has been long viewed as a caricature of novel tissue renewal. So it can occur almost any part in the body, and it involves complex interplay with the surrounding tissues and forming an ecosystem. So systematic approach can really help us to understand or transform the way we understand the mechanisms that drive cancer progression and how they respond to treatment. In addition, we can also learn from the development, and especially when we study the dynamic evolution of the tumor ecosystem, and especially for the treatment-resistant case, and to see how they change for different populations, both the tumor and the microenvironment over the course of different treatment. So both development and cancer are very complex processes. However, we can organize these into different axes to help us understand the different layers of molecular regulations and the cell heterogeneity and also evolutions across time and the space. Coordinate multi-layer gene regulation played critical roles to control the cell phase and at different stage and control the key cellular transitions. So actually, instead of to avoiding the complexity, we can actually harness its potentials. So we can actually look at and integrating different gene layers, including upstream gene regulations and also downstream targets, and tracking these change over different time and also spatial, as Sheila mentioned. And that's actually help us to better understand the genotype to phenotype relationships and identify key driver event versus noise. So as a system biologist, and I'll discuss how we can develop different technology platform to help us to understand these questions and answer the questions in a timely manner. So just a very quick outline for the talk. I'll stay on the brain context but highlighting two stories. One is the novel development, but the other one is a brain tumor glioblastoma. This will further lead to the discussion of some ongoing broader applications and collaborations in the lab and also with the broader community. So as I mentioned, brain is one of the most complex organs in our bodies. So during brain development, there are multiple waves of gene regulation that control cell phase, such as from neuroepithelial cells to progenitors and the different types of neuron and the glia cells. At the same time, when the brain size significantly increases during development, it involves very complex cellular organizations and interactions. So it's actually very important to study this spatial and temporal change. And on the other hand, so brain tumor is actually represent a dysregulated developmental processes. So it's representing a complex and dynamic ecosystem and involving diverse cell populations and the states, and also different cell population from the non-malignant compartment, including immune cells and different brain cells, and all these cells actually interacting together and during the disease progression and treatment response. So it's important to study these at different stage. So as I mentioned, in addition to individual layers, we're also interested to see how different gene regulator layers actually connect together to play an important role. And I think every of you might be having your favorite layer of gene regulation or using alternative splicing as examples. So using RNA sequencing from a large panels of tissues and the cell lines, we actually identified MBL proteins is lowly expressed in pluripotent stem cells compared to all other different cell lines and the tissues. And importantly, we further show MBL1 and MBL2, which are RNA binding proteins, and they are function as a master negative regulators to control a very large program of stem cell alternative splicing networks. And this event actually can regulate different gene pathways, and including chromatin regulators and also key transcription factors such as OCT4 and the NANOG. And this provides the first evidence splicing factor in the stem cell context actually can function as co-regulators and coordinate different layer of gene regulation to control stem cell pluripotency and also iPS cell reprogramming. So not surprisingly, so MBL regulated alternative splicing program also being shown in different types of cancers. Low expression of MBL have been indicated in brain cancer, gastric, and breast cancers. So the low level of MBL is also associated with poor patient survival and also increased relapse and metastasis. So if we're looking more broadly, and here is on the top, is showing different types of alternative splicing events, and it can actually impact every hallmark of cancer, such as apoptosis, proliferation, or even immune response. So some of you might know, actually, more than 95% of human genes undergo alternative splicing. So if you look at your favorite gene of interest, and very likely there are different isoforms. So as mentioned, so by studying alternative splicing instead of avoiding the complex, they actually give us the opportunity to discover new biology and the therapeutic targets. So there are actually three connected components for many of our research programs in the labs. So typically we're starting with the pressing questions. So for development or cancers, and these biomedical questions actually is the driving force for us to developing new technology platforms, especially when the current approach having limitations. And so in parallel, we also develop our bioinformatic pipelines in addition to technology platforms, which further help us to accelerate discoveries. And importantly, we're actually using high-throughput studies and as a hypothesis generation process. So this further leads us to identify key biomarkers and also therapeutic targets for in-depth function and the mechanism characterizations. So this one will further lead into additional development of technology to forming a systematic discovery cycle. So for the next part, I'll discuss how we actually develop and employ a highly multiplex functional genomic screens and also single-cell profiling to identify a large networks of key regulators to control neurodevelopment at different stage. So as I mentioned, if we look at in different regions of the brain, there's actually specific gene regulation programs, including both gene expression and alternative splicings. And also, when we look at things through times, there are specific dynamic patterns regulate over different time points of neurodevelopment, increase, decrease, or even more dynamic change over the process. However, to study this spatial temporary regulate neural alternative splicing event and expression event is quite challenging. And if we look at in each of these individual events, one by one, it requires tremendous amount of work. But also on the other side, we could be easily missing the big pictures by looking at single events and not the chronic regulations. So to better understand multi-layer gene regulation in a complex system such as brain development or brain cancer, I developed a highly multiplex functional genomic screen called SPARSIG. So just very briefly, and so actually we developed a highly multiplex RT-PCR reaction. So this allows us to monitor hundreds of endogenous regulation events in a single reaction. So you can monitor expression, alternate splicing, non-coding, or other RNA regulation events. And so you really can be creative for the panel designs. And then we actually design a flexible barcode system, and then we can use it to tracking different panel of events. And by using this barcode, we can track different samples or different treatment. So then we can put in all the samples together, thousands of them, and then to sequence them. So this really gives us a highly efficient and cost-effective system. We can study a large panels of an event together to systematically discover genetic regulators, but it can also couple with chemical screen to identify important therapeutic reagents. So at the same time, we're also creating a lot of resources. I'll circle back later. So now with this platform, we can really analyze all the spatial temporary regular events we are interested in one study. So as I mentioned, we can actually design our sparsing panel event to including different types, for example, expression, splicing, but also selecting different subcluster or dynamics events at different time or region specific, and including control event as well. So in that way, it actually can help us not only identify a list of regulators, it also provides insight right away from a screen. What is the specificity? What is the potential mechanisms? And even the physical or functional relationships among different regulators we identified. So from a very in-depth readout and just from a single screen, we actually can identify hundreds of positive and negative regulators important for different stage of brain development. So we actually recently published this study, and it's a truly amazing teamwork. So please read and if you're interested for this. And however, we don't stop there. And sorry, apologize for this version of the slide. It's overlapped. So actually, we don't stop there. We can actually further integrate sparsing information with another molecular information. An advantage for this is, so for sparsing, we can merge in different screens. For example, the neural screens we perform, but also a stem cell screen. And also with the sparsing team, we further integrate the data with the bulk and the single cell RNA sequencing during in vitro differentiation and also in vivo development. So by generating these multi-model integration networks, and it actually help us to understand the specificity and the mechanisms and selecting the top hit for a downstream function and mechanism studies. So for example, we discovered negative regulators in one cluster and also positive regulator for neural regulation and the splicing regulation in one. One thing we found is very interesting is some of the factors we identified, they show a specific neural expression patterns low or high. But there's also a group of clusters we identified. They are uniformly expressed in different cell types. So we wonder how they can achieve a neurospecific regulations. So one example is POP60. So what we're looking to further of POP60, we actually found there's an exon 5 adjacent to the first RNA binding domains, and it's actually highly conserved during vertebrate evolution, and importantly, it's neuroregulated. So first, the other factors, which is a neurospecific, we can simply knock down. But for POP60, we actually want to more accurately modulate the isoforms. So using CRISPR, we can specifically delete exon 5 or having the HDR CRISPR to help us to inserting a CD version. So exon 5 is always included. So make a very long story short, and we're actually performing different assays to really understand the mechanism and the functions of POP60 isoforms. So we study the morphologies and also performing bulk RNA sequencing analysis for both expression and also alternative splicing, performing AP mass spec to review the protein interaction networks for both POP60 isoforms. So importantly, I just want to highlight, and based on our observations, so the minus exon 5 is critical for early stage of neurodifferentiation from stem cell to progenitors, while the plus isoform is important for late stage of neurodifferentiation from progenitor to immature and mature neurons. But it's also leading to the next questions, whether POP60 isoforms have an important role for cell type and the different developmental stage role. And so we really want to look at single cell resolution. But in order to analyze different genotypes, we really need a highly efficient system. So I'll discuss more for the single cell platform we build and using that. But here, just make it simple. This is a single cell cellular maps, and we're showing after integrating different genotypes and also different types with profiles. And with the platforms we build, we're actually able to analyze a large list of samples together in a single experiment. But from the single cell maps, it really can help us better understand the cellular heterogeneity during in vitro differentiation, but also the trajectories from stem cell to neurodifferentiation. And also I want to point here, in addition to single cell cellular maps, we can also creating gene regulatory networks from the single cell data, such as the plot shown on the top. So each of the dots is actually showing a gene instead of a cell in the bottom plot. So we can actually see the gene programs that are specifically regulated in different cell populations. I only highlight two examples here. For example, the EMP and the ECM pathways are specifically associated with a rare population of non-neural cells in the differentiation, and they are like a vascular-like and but also we can actually further link this change to different genotypes. That's what we're interested in. So here again, we can see the EMT and the ECM pathways is actually more upregulated in the minus exon 5 isoforms, while neural pathway is more regulated in the plus isoform. So in addition, we can also checking at cellular abundance levels, and this further confirmed the observation that at a later stage, plus exon 5 is promoting more efficient neurodifferentiation by inhibiting this non-neural cell population. So quickly summarize these parts. So we have developing a neurosparstic screen and also integrate with the single cell to really help us to understand the different stage of neurogenesis. And this really helps us to identify key regulators. I think POP60 I showed you is just one example. So from the screen, we actually discover hundreds of factors for future development and the disease studies. And also, sometimes we found actually many of the factors we identify not only play an important role during neurodevelopment, but also disease states and different cancers. So for the second part, and our shifting focus to kind of present our study for glioblastoma, but focusing on how we can use systematic single cell characterization to help us further understand the mechanisms that drive GBM progression and the recurrence. So you're already hearing the amazing story and learn a lot for GBM, so I'll keep the introduction really brief. So GBM is the most common primary brain cancer in adults, and so even with maximum therapies and almost all patients experience relapse and in a very short window, as you hear from Sheila. And it's also the most lethal and heterogeneous. And so it's really the whole ecosystem of GBM, and it's involving different tumor population, but also a lot of different immune cell population, and especially in the brain context, the brain cells play an important role as well. And the GBM cell population even can hijack the novel neurodevelopmental program as a way to contribute its own cellular heterogeneity and plasticity. And also both the GBM tumor and microenvironment is rapidly evolving during the disease progression and the different treatment. So that's why at recurrent stage, we actually look at a very different tumor and microenvironment comparing to the primary stage. So to really better understand the mechanisms and develop effective therapies for GBM patient, and we need to look at this evolving GBM ecosystem at different stage. So in addition to patient primary and recurrent GBM, and we're also analyzing the end stage of GBM disease and with different tumor and the brain regions. And this is in collaboration with a large GBM teams and also our recent established rapid autopsy program at McMaster by Dr. Sheila Singh. So internally, actually using the high throughput platforms we built, we actually want to creating a more ecosystem view to understand the dynamic interactions between different cellular populations, tumor cells, immune, but also give us to see the brain cells interactions. And this can help us to more comprehensively understand the molecular mechanisms that's driving the GBM progression and the treatment response, and help us to identify novel modalities as well to kind of developing prognosis, diagnosis, and the therapy. So you can imagine to analyzing such a large cohort of patient samples across different time points and the regions, we need a highly efficient single-cell profiling platforms. So many of you probably hear or use 10-time genomics as a way for single-cell profiling. So this instrument actually creating a droplet, so you actually having a cells and also barcode bead in the droplet to uniquely label transcriptome from each cells or nuclei. But we're asking the way to see, is there other ways more efficient and cost-effective? We can also uniquely label the molecular information from each single cell. So actually, we can using the nature nuclei or cells as a compartment to label the transcriptome or other omics. And this cool concept is called single-cell combinatorial indexing. And this is also the method we use in the lab to really building an ultra-high-throughput version of single-cell profiling. So the really key for this method is multiple levels of barcoding. So for example, we can introduce the first barcode using a reverse transcription reaction. And each walls is getting cells a different barcode. And then we putting all the cells together and splitting to the second levels of a barcode. And then we repeating this pulling and splitting again. And this is really give us a huge combinations from three levels of barcodes. And each level we actually using 384 different barcodes. You can also imagine this as a birthday question. So if we kind of founding from this audience and having two persons having exactly the same birthday date, it's possible. But if we want to founding a group of three people exactly having the same birthday, it's going to be really rare. So that's how we're using these combinations of barcoding to achieve a unique label of the molecular information for each cells or nuclei. And I'm also very excited because it's really giving the flexibility. We can do different omics instead only transcriptomics levels. So using this approach, we're actually building our own in-house ultra-high-throughput single-cell profiling platforms. So the skill and also the capacity of experiment we can perform is a significant increase over time. So as a quick example, even just with 3 or 4 people as a team, we can easily profiling more than 1 million of single cell or nuclei from a single experiment. And so this can really help us to analyzing a large cohort of patient samples and with different time and spatial comparisons. But I think the key here is because we're actually developing this technology, we can actually generating this sequencing library in the way we want to help us to understand the body. So for GBM case, we're interested to get an ecosystem view, but also in certain studies, maybe you only want to analyze in T cells, but actually across a very large cohort of patient samples. So using these platforms, it gives us a highly efficient and cost-effective approach. And also we're moving the further upgrade from single-cell RNA sequencing to a single-cell multi-omics. And in parallel, we're also developing our own bioinformatic pipelines to helping more efficient discovery. So using this approach, actually, we have two parts of analysis. Both are very important. So starting with the raw sequencing rate, and we need to demultiplexing this rate and mapping to different genes or different isoforms, and then using this gene to cell matrix, and we can performing downstream single cell analysis. And for different single cell analysis, we'll be developing more than 20 modules. And so here are just a few examples of the basic modules, but we're going to continue developing these analysis tools and share with the community. So using a pilot study as examples. So from this study, we're actually profiling 18 patient-matched primary and recurrent GBMs, and in collaboration with large GBM teams. And so from this experiment, and because we want to analyzing different cell population from the ecosystem, so we don't performing any cell sorting. And so after single cell profiling, the first task we need to do is to separating the tumor and the non-tumor population. So here we're creating a multi-level scoring system, so including copy number variation analysis, GBM and the normal brain cells marker identifications, and also unsupervised clustering. So this really helps us to be more efficiently and robustly to distinguish tumor and the non-tumor populations. So when we look at the tumor cells, the first thing we done is doing a gene marker analysis and also pathway analysis. Showing here, you can see we can clearly distinguish primary and recurrent GBM, and this is a good indication. In addition, we also look at in over 100 different pathways that are important for GBM biology and to discover. So one observation we made is actually the recurrent tumor is more proliferative and also less response for immune. So this is partially expand why GBM at the recurrent stage is harder to treat. But on the other side, we also look at the non-tumor populations. And interestingly, we actually found many of the non-tumor population, especially certain brain cell population, is a significantly more abundance at the recurrent stage, and such as the oligodendrocytes, Sheila also mentioned. And actually, we're already making cross validations using independent data sets and the cohorts, and currently we're performing additional function assays to study their functions, including spatial transcriptomics and also co-culture assays and the in vivo models. So in addition to patient samples, we're also interested to characterize and the different in vitro and in vivo models. And we want to integrate patient data and with disease models we're creating to founding the consent regulators and especially the key driver event to guide our downstream studies. And we, for example, we're creating different knockout cell lines and inject to the mice and as a tumor model. Just give you one example. So with this ultra high throughput single cell platforms we built, and in a mouse model, we're actually able to look at in a whole brain regions. And that it gave us to opportunity to really build the interaction networks between tumor, immune, and the brain cells. And using one of the knockout models, and we actually from a pilot observations and the knocking down of RNA binding protein candidates we discovered in the pilot study, it's actually when we knock down it, it's actually increased the survival of the mouse. And right now we're actually analyzing the single cell data to really understand the function of these RNA binding protein candidates. So far we have building this ultra high throughput single cell platform to help us to understand the patient GBM samples at different time point or the spatial heterogeneity samples. And also importantly, it's using this approach, we want to gain an ecosystem views to discover novel biomarkers and the potential therapeutic targets. And this is like a teamwork, and I think by combining expertise from different areas, and it's really help us to identify the novel target at the gene level or isoform levels in order to further improve their therapies we urgently required for GBM patients. So for the last part, and I'll just to see the GBM story I discussed is one of the research projects in our team. And with the established collaborations and also our GBM team, especially Sosa, Kabir, Vanshika, and Alberto, they have been further developing the single-cell platform to help study their patient-matched GBM samples. Here I just listing two priorities, and we have recently upgraded the platform so we can actually performing co-analysis of gene expression, alternative splicing, and mutations at RNA level at single-cell resolution. This is really help us and with the different layer information to further understand the GBM heterogeneity and also the combinations of different molecular layers to driving the GBM progression. And also this year, we're going to add in single-cell epigenetics to our platforms, especially single-cell ataxic. But also we are prepared for the next stage of development as well. So the platform we use is also compatible with additional single-cell omics, such as meso-seq, high-seq, and also spatial omics. At the same time, we also developed multiplex screens, sparse-seq or perturb-seq-like screens, and to really help us to more systematically identify key genetic regulators for different genes and isoforms. And also, we actually developing therapeutic screens using these multiplexed screen as well, so it can help us to quickly screening thousands or millions of drugs and in a very quick way. So we're actually also developing such platforms in different formats. So in one version, and using 96 format, we can actually purify total RNA upon different genetics perturbations or chemical treatment, and creating a sample library. And our sparse-seq was designed in such a way, each purification is enough materials we can use for 20 and 30 screens. So following the initial panels of discovery, we can also further design new panels of events to identify regulator and integrate the data to giving a more understanding of their specificity or mechanisms. So as a quick example, the sparse-seq teams and listing here and actually we collaborating with Dr. Hansen, her team, and just finishing a pilot screen for treatment-resistant neuroendocrine cancers and to discover regulators for both splicing and the non-coding RNA regulation events. So for the last part, we are also in general interested in coordinated splicing regulations at different cancer stage, initiation, progression, and the treatment response. And also we generating internal resources of more than 3,000 RNA binding proteins, and we can look at the RNA binding protein that express or detect from our single cell data to really look at alternative splicing and its coordination with other layers at the single cell resolution. So I think from the program and the research project I shown you, and you notice we always closely integrate cancer biology with technology development. And the framework and the technology platforms we built can be easily applicable to different cancer types and the models. And as a member of CDCR and the cancer, the Center for Discovery of Cancer Research at McMaster, my team is particularly interested in treatment-resistant cancer and also unique patient cohort comparison. So for example, the spatial temporary GBM patient samples I just showed you, but also another example is a local curable versus treatment-resistant prostate cancer. But it's also really nice to having the open discussion and hear from you what will be the samples and the question you are interested in. And in addition, we also building and characterizing different models for systematic function and the mechanism studies. And when we're building these technology platforms and the bioinformatic pipelines, we're also creating a lot of resources. For example, for the single cell pipeline, we're having a very large barcode primer and for multiplexing systems. And during the SPARSIC, as I mentioned, we can also creating sample libraries and for collaboration to screening different panels of the event. However, what one team and one center can achieve is always limited. So it's actually nice to kind of see what we can achieve in the next stage as a larger team. So in collaborations with us clinicians and the fellow scientists, we're also interested to integrate clinical information with single cell profiling and the in-depth screening data we generated. So this can help us to creating a digital biobank. And using this one, we can actually, for our collaborating teams, but also broader community to discover novel modalities that are important based on different levels of information. So I think slightly in a longer term goal, and it's in the next stage, we're going to see further integrations of different areas of cancer research, and also to see how cutting edge technology can help us to, for both basic and translational cancer research. And using single cell multiomics as examples, and it's really already helped us to directly profiling patient sample in depth and also characterizing and validating different cancer models. And by using the multiplex screening strategy, either as bulk or single cell, we can actually identify genetic regulators in a more efficient way, but also real-time therapeutic screen to help identify therapeutic targets and developing reagents. And in the near future, I think we're also going to see it's a further coupled with clinical trials. And so we can actually learning critical insights, not only from the successful case, but from failures to help us to develop novel, more effective cancer therapeutics. So actually, as an early career scientist and working in the field, and I hugely benefit from these interactions and learn so much from our collaborators in different fields and to better developing cancer models and understanding cancer immunology, drug discovery, and AI, and also from our clinical collaborators, we learn more about our need from the medical side, and also what patient samples and clinical information we can further integrate from the molecular informations. And it's exciting to see the next stage for this ongoing growth and the development of a collaborative enterprise. So just acknowledgement as huge thanks for all my collaborators and also mentors and across different projects and across my career path as well. And the two teams working on the two different projects and also my current teams at the McMaster. And thank you for your attention.